Thank you. So everyone, hello, and thank you for attending this talk. Today, I will be talking about a paper that we published recently, early this year in the A-Life journal, and it's titled "A Spatial Artificial Chemistry Implementation of a Gene Regulatory Network Aimed at Generating Protein Concentration Dynamics." So our work is based on an earlier work done by Dr. Wolfgang Banzhaf in 2003, and the work was titled "On the Dynamics of an Artificial Regulatory Network." And the objective of our study is to introduce a more biologically realistic and a spatial model of gene regulatory networks, or GRNs. It's also to analyze the impact of initial states of this system on the dynamics that are produced, and to investigate if we add stochastic elements to the system through the movements of artificial agents in a 2D grid, that would result in a robust and evolvable system that can produce evolvable protein dynamics. And also, finally, to explore potential applications of this system. So just to generally recap, about gene regulatory networks, we know that differences between species depend on timing and intensity of gene expression. And GRNs are basically complex biological networks that are in charge of controlling gene expression and protein production. And just to simplify the gene expression process, it's consisted of many entities, but maybe like some of the important ones are the promoter region on the DNA, which basically indicates the starting point of a gene and the starting point of transcription. We have enhancer and inhibitor regulatory sites that increase or decrease the likelihood of transcription. And we have the transcription process, in which DNA is turned to RNA, and also translation, in which we build proteins out of the RNA. Some functions of GRNs are cell differentiation, metabolism control, signal transduction, organism development, and so on. And it's important to understand GRNs because it helps us understand DNA's role in life, and it has application in genetic disorder identification and treatment, and also they can be used as computational tools. So the proposed GRN model in this work is a spatial model, but it's based on a linear DNA sequence that is not really spatially modeled here. It has a 2D grid, as you can see on the right side. This is a picture of the initial state of a GRN system in our work. The system is agent-based, means that we have different agent entities in the system that can interact with the grid and also with each other. Interactions between entities are basically formulated using artificial chemistry, and we have some stochastic elements added to the system through the movement of these agents in the system, which is random. We have the notion of time, which is basically cycles of the system, and in each cycle, different phases happen, different entities do their job, and they interact and change the dynamics accordingly. And the production of this, or the output of this model, is protein concentration dynamics. So if you look at the picture on the right side, these squares that are spatially represented on the four corners of this square are transcription factors of the GRNs. The triangle shapes are enhancer regions, and the circle shapes are inhibitor regions. And the color coding means that these are from different genes. So this is the enhancer and inhibitor of the same gene, and these are for different genes. And then if we see a transcription factor with the same color, it just means that the protein or the transcription factor is the product of that gene with the same color. And then the final output of this model is the protein dynamics, which are interesting for us. This picture shows our model in 3 different stages. This is the initial stage, or cycle 0, in which we have transcription factors far from the regulatory sites on the corner of the square or the grid. We have early stages, so some cycles have passed, and the transcription factors have moved around a little bit randomly. And then on the later stage, we can see that the transcription factors are uniformly spread in the 2D grid. and they can just interact with the other regions if they become close enough. For example, this red transcription factors is binding to like this triangle, which shows the enhancer region of that gene. So as I said, everything starts from a linear DNA sequence that is not spatially represented. So how do we find these genes that we're talking about? How do we make this like 2D system out of the DNA sequence? So the first things first, identifying gene. And the way that we do it is that we look for a fixed promoter sequence, AGCT in our case, in the DNA, and that represents the start of the gene. And then we keep searching for a terminator sequence, which is also fixed in our system. And once we have found that, and it has like at least some sites in between, so it has like a small limit size, minimum size. And once we have found that, then we can go on and identify different regulatory sites of that gene. So everything that comes between the promoter and the terminator is basically links of the gene in our case. And the size of the regulatory sites, meaning enhancer, inhibitor, and also the genetic markers for the protein produced by this gene, they are the same in our system, and it's calculated by getting the square root of the size of the whole gene. So S is size of the enhancer, inhibitor, and the protein marker, which is calculated using the protein coding region. So how do we calculate the genetic marker of a protein? The enhancer and inhibitor come right after the promoter, so we don't have to actually locate them. Like it's simple to locate them basically. But the protein region, the genetic marker of that, we have to calculate it from the protein coding region. And the way that we do it is that we divide it into different number of chunks, n chunks basically, which is the same as the size of the other regulatory sites. And then we use a majority rule for each of these chunks to finally calculate the protein sequence that this gene would create in our system. I talked about transcription factors moving around in our system and binding to different regulatory sites, but the binding actually happens following this rule. So we have different transcription factors. They can have various sizes. It doesn't matter if the sizes don't match. And we have regulatory sites. So when we have a transcription factor, we use a binding rule, C binds to G, H binds to T, no other base pairs bind. And basically based on the number of bindings, we know that how strong the binding is between the transcription factor and the gene. And then we have some exponential formulas that we use for determining the transcription rates and also how to update the concentration levels based on these transcription rates. And this is calculated for all of the transcription factors that are connected or bounded to the regulatory regions. So in each cycle, we have some phases that happens. First, the transcription rates update depending on the TF bindings. And then once we have that, we can update the transcription rates accordingly. There's this movement phase in which transcription factors do a random walk in the grid. There's this binding phase in which if the transcription factors are close enough to the regulatory sites, they bind. There's the production phase in which the concentration levels of the proteins are updated. And then lastly, if the binding duration between a TF and also a regulatory site is over, then what happens is that we remove that transcription factor and we create a new transcription factor. At the start of the system, we have the same number of transcription factors allocated for each gene that is present in our system in the 2D grid. So let's talk about some experiments that we conducted using our system. These parameters that you can see here are the general parameters that we use for most of the experiments, unless we stated otherwise. So the number of cycles is 1,000, so that's like 1,000 time cycles for each model that we have. Then we use this grid size, the number of transcription factors. As I said, it's the same for all of the genes at the beginning or throughout the system. And then the starting concentration level. So we don't start from zero. Usually we start from the same concentration levels for all of the proteins that are available in our system or can be produced in our system. Then there's the step size for random walk. There are two parameters that we use in our formula that are basically arbitrary parameters that control the production rates. And the DNA length that we used, and this is the DNA length, basically means that the DNA sequence that we randomly generate for each of these models, and then we identify genes from this DNA sequence. So the whole length of that is 3,000 bases. So once a system is randomly initialized, it can produce varying protein dynamics. So we basically classify the different types of these protein dynamics into different categories that kind of like resembles a particular movement. For example, we have the simple development dynamics in which we can see that the movement of the changes in the levels of concentrations are basically steady. They don't change too much. We have oscillatory behavior. We have hybrid behavior in which we have oscillation, and also we have simple development. And we have the chaotic behavior in which it's hard to determine the pattern that these protein dynamics produce just by looking at the dynamics for like a portion of the simulation. For example, 1,000 cycles. It's hard to determine what's happened next. So the left picture here shows a protein concentration dynamic. And then on the right side, we're particularly looking at the transcription rates of protein 5, which is the purple one on the left side and also here. And as you can see, there's a correlation between the two. For example, at first, there's a high transcription rate. Then you can see the jump in the level here. And then as the time progresses and the transcription rate is very low, the level is just reducing. And then you can see the oscillation here happening and here also happening at the same time. The next thing that we tried was to change the initial states of the system and to see what sort of impact it has on the produced dynamic. So for these experiments, we have the same system, we have the same DNA, we have the same genetic markers for everything, but we're changing just a single value that is an initial state, basically. So this is a base dynamic, and for this case, we changed the beta and the delta values. So this figure that you can see is just for the beta value. We increased the in slightly, and we could see that for one particular protein here, which I think is protein 1, we can see that shift in time happens. The same thing happens for all of the proteins, but it was too just too crowded to just include it all in just one figure. So I just included protein 1 changes, but it's the same for all of these. This is for the case that delta value is changing. And then again, we can see the time shift happening, but it's not as big as the time shift that you would see for the beta value. And yeah, like it kind of makes sense if you look at the formulas that we use for the transcription rates. But it gets more complicated when we get to the things that are not so trivial. For example, changes to the initial concentrations that we have in our system. So we, as I said, we don't start from 0 concentrations for the proteins that are produced. We start from different values, but this is for the case that we start from the same values for all of the proteins. And this is for the case that we have the same system, the same marker, the same DNA, but we're starting with random values. And you can see that the dynamics produced by the system highly depends on the initial state, and it's somewhat different. The next thing we tried was changes to the initial transcription factor counts. So what if we have more transcription factor counts for each gene? Maybe you have like less. Then in that case, we can see that, again, like time shifts happening. The time shifts are not really as consistent as you would see like with the changes with the beta on the beta or the delta. They're a bit more random. Like these are like more consistent, but this is like a bit more random for transcription factor counts. And it makes sense because transcription factors randomly walk in the 2D grid. But it also shows the robustness of the system because the whole dynamics do not change completely when something like that changes in the system. The other thing that we tried was to see what if we have different number of transcription factors for different genes, then what happens? So this is for the case that we have the same number of transcription factors, and this is for the case that we have added only one extra transcription factor for one protein, which I think is protein 1. And then you can see the dynamics are changing. And you don't see the concentration levels of 5 proteins here because some of them are overlapping right there. The next thing was to change the grid size. So what if the cell size or the grid size is bigger, then what kind of changes can we expect? And again, we can see a time shift happening. It just means it kind of resembles the fact that if the cell size is bigger, it takes more time for the transcription factors to uniformly spread in the system and produce the dynamic that is more stable. And then we targeted changing spatial properties of the system and to see how much of an impact it does. And one of the most impacts that we got from changing initial states was doing a spatial mutation. And what we did was that this is the base dynamic on the left side. And what we did was that we changed the enhancer region of one protein, the position of the enhancer region of one protein just a little bit. As you remember, like in the 2D grid, the transcription factors move around randomly, but the regulatory sites, the inhibitor and enhancer sites do not move. So what if we move them just a little bit, just one of them, and we can see that the whole dynamic drastically changes. So the last experiment that we tried was to see if we can evolve these dynamics, and we tackled 2 problems. One was to target a certain concentration level for a certain protein at a certain time cycle, and we tried to reach that value basically, which is marked as this red circle right there. And we noticed that we can evolve such system in less than 20 generations that can solve this. We can like evolve this dynamic, and we only had like 10 replicates here, so nothing magical was going on the evolutionary side. Then we tried to evolve this oscillatory behavior, and we had this scoring mechanism that would force something like this. It was a bit harder task, but then again, in 25 generations, we could solve it, showing that the system can be evolved. And this slide is a bit too complicated, but all I want to say is that we can have different solutions for the same problem. So it doesn't have to be necessarily the shape that we can see here. And finally, we measured the impact of mutations on the produced dynamics. Sometimes mutations are neutral. They don't change the dynamics at all, but sometimes, so for example, mutation, when there's no mutation and there's one mutation, the dynamics are overlapping. And for 2 and 3, we have it here, and then like we have it here, so we can see changes in the system basically. And 4 also overlaps, I think. Just to wrap up, what we did in our research was to present an agent-based GRN model using a 2D grid and an artificial chemistry. We noticed that the protein dynamics that are produced by the system are similar to the biological counterparts, and we explored the impact of the initial states on the system. Excuse me. And we highlighted the robustness and evolvability of the system. Some of the key observations that we noticed was how we can control the system by changing the initial states, how the slight changes to the spatial positions changes the dynamics, and how we can use like evolution to come up with different solutions for the same problems, and how mutation impacts the evolvability. And the future directions of this work is to test GRN representations or artificial regulatory network representations for genetic programming to solve complex computational problems. And one thing that we thought about was to introduce spatial genes to introduce inputs and outputs to our system, and to perform quantitative analysis on the dynamics of the evolution rather than just observations, and to study the impact of the DNA size and the number of genes on the complexity of this network. So thank you very much for listening to me, and I'm here if you have any questions.